congratulations. Okay, this whole blue thing is our cytoplasm, and we have our mRNA. This mRNA, the full name actually would be what? It's going to be mature mRNA. mRNA. So that means it's been gone through the splicing, okay, and all the non-coding introns are taken out, and it's got only coding, okay, only exons. So this is my mRNA. It looks like a wiggly line under a straight line. This is my mRNA. So let's. Okay, the mRNA is gonna go in the cytoplasm. In your cytoplasm, you have lots and lots of these small dots that you see on a microscope. We call these ribosomes. Ribosome, I want to magnify it. Ribosome is basically, let me use this as a ribosome. This is my ribosome, right? The ribosome is going to attach itself at the first part of the mRNA. That makes sense, first part. So I have a ribosome sitting here. All right. So obviously, and in here I have my bases. So let me just do them. A, T, that's wrong. Why? This is RNA. Should be U. U, G, C, C, G, and rest I'm just making it up. I'm making sure that I put no T. Okay. Oh, another T. A, C, G, G. All right. This is my copy of my code, mRNA, mature mRNA. So my ribosome is going to sit here. Okay. So over there, I have lots of cytoplasm. So lots of amino acid. This is my amino acid. My amino acid's name is MET. MET is the amino acid, MET. But we said the amino acid is not by itself. It's attached to a tRNA. So this is my tRNA, and it's attached here. This area is called amino acid binding site. Now, then I have 3 bases here, okay? These are called anticodon. So if I want, so in here, AUG, 3 of them is codon. If you remember, 3 base is codon. I have 1 codon here, another codon here, another one, another one, another one. So I have 1, 2, 3, 4, 5 codons. Each codon generally codes for 1 amino acid. So if I let's do one more amino acid. I have serine amino acid, and this amino acid is held by a tRNA, and this is my tRNA, and you have these 3 bases, anticodon. AUG, which amino acid should it code for? This one or this one? We don't know. But we do, okay? If it wants MET, these bases means what? UACG. Any of these 4 that will be here. They have to be complementary to here. So if I want MET, this should be what? A, U. U, A, G, C. These letters, these bases are anticodon, meaning they are complementary to the codon here. Let me do another one here. Maybe GGC. GGC. So I have 2 amino acids. You have 2 options. Should it bring this amino acid or this amino acid? And you will go by which one is complementary. Which codon is complementary to the anticodon? This is what's called anticodon. It's the opposite of the codon. So we know that GGC is not complementary to this. This one is. So this tRNA is going to transfer the amino acid, move all the way and come here. So over here we have our tRNA, and this is the anticodon, UAG, UAG, complementary base pairing, codon, anticodon. This is my tRNA, obviously, and it has brought my amino acid, MET. Job done. the amino acid has been brought. Then what happens is, I know that I have more amino acid to bring. So the next amino acid, so next codon, so CCG, you have so many amino acid. So this one, GGC, is complementary. You have other ones which are not complementary. So this one comes all the way and comes here. So GGC. This is my anticodon, complementary. I have my tRNA and I have my serine. This is my amino acid. I have two amino acids together. The order is correct. Codon, anticodon, complementary. Now you have an enzyme that also catalyze a reaction between these two amino acids, making a peptide bond. The ribosome is quite small, so I made two amino acids. The ribosome can only fit two codons. So ribosome can fit this codon up to here. So now my next part is this one here. It can't fit. Ribosome now does two amino acids and then it moves one more. It can only fit two codons. So the ribosome moves one more and the next one occurs. The next one brings the amino acid here, complementary codon, anticodon. So you have another anticodon brought by tRNA, you have another amino acid, and then you have a bond there. Then the ribosome moves again, and again, like that. The last one here, this over here, this is your last amino acid. You might think, how is it last amino acid when I have one more codon, correct? How is that my last amino acid when this is my last codon? This codon, the last codon of mRNA, is called stop codon. It tells the ribosome to stop here. This is your last amino acid. There's nothing left. That's the end of the protein. So we always have a stop codon right at the end. At the beginning, AUG, this is our starting, right? We call this start codon. So let me recap. The first codon is called start codon. It does code for amino acid, methionine. The last codon is stop codon. It does not code for any amino acid. Big difference. So then what you have your amino acid joined together, primary structure. Amino acid is now a protein. So going back to your first module, you have your primary structure, then secondary structure, hydrogen bond, and then tertiary structure, and so on. mRNA has done its job. It just gets broken down and destroyed. No need for mRNA to be done. And the protein is made.